In the video lesson entitled Overview of Heredity Topics, you have learned that meiosis plays a significant role in gamete formation, as well as in producing variation among offsprings, partly due to the exchange of genetic material between homologous chromosomes, in the event called crossing over. In this video discussion on meiosis, we will look more closely into the chromosomal events that take place in each stage of meiosis. A big chunk of the discussion is devoted to the chromosomal events that take place during prophase 1. It is also expected that after watching this video, you should be able to explain the significance of meiosis in producing variation, and in maintaining the chromosomal number of an organism. Meiosis consists of two phases, meiosis 1, and meiosis 2. Meiosis 1 is also called as reductional division, because the chromosome number is reduced into half. On the other hand, meiosis 2 is also referred to as equational division, since chromosome number is maintained. We will look more closely into the processes of reductional and equational divisions as we go along our discussion. At this point, keep in mind that the reduction of chromosome number into half is of great importance in the maintenance of chromosome number among organisms. Let us now discuss the chromosomal events that take place in each stage of meiosis. We begin with the events that take place in prophase 1. In prophase 1, the nuclear membrane begins to disintegrate, and spindle fibers begin to form and migrate towards the opposite poles of the cell. The chromatin material also undergo coiling and thickening, to assume the appearance of chromosomes. At this point, we will be delving deeper into the 5 subphases under prophase 1, and the chromosomal events that take place in each subphase. The initial coiling and thickening of chromatin material into chromosomes take place during leptothene. Chromosomes continue to thicken as they progress to the next subphase, which is zygothene. In zygothene, homologous chromosomes pair off in the event called synapsis. It is apparent in the illustration that a pair of homologous chromosomes consists of 2 members. One is of paternal origin, while the other one is of maternal origin. At this stage, a pair of homologous chromosomes is also referred to as bivalents, simply because each pair consists of 2 chromosome members. Homologous chromosomes will assume their thickest forms during the pachythene stage, after undergoing further coiling and thickening. Owing to the thickness of the chromosomes, the individual chromatids that make up a pair of homologous chromosomes are distinct and identifiable. As we can see in the illustration, 4 chromatids make up a pair of homologous chromosomes, so we refer to these as tetrads. In the next phase called diplothene, the non-sister chromatids of a homologous pair overlap. This process called crossing over is the exchange of genetic material between members of a homologous pair, which will eventually lead to variation of traits. The specific region along the length of a chromosome, where the overlap or crossing over took place, is called chiasma. Its plural form is called chiasmata. That means, it is possible to have 2 regions of overlap between members of a homologous pair. Members of a homologous pair will be held together at the chiasma, or region of crossing over, as they separate or move apart from each other, in the last stage of prophase 1, called diakinesis. Terminalization refers to the movement of chiasma, towards the ends of the chromosomes. As you can see, the paternal chromosome have acquired a segment of the maternal chromosome, and vice versa, which is an indication of the exchange of genetic material between the 2. It is during the last stage of prophase 1, that the nucleolus disappears, nuclear membrane disintegrates, and spindle fibers begin to form. During metaphase 1, the chromosomes align themselves along an imaginary metaphase plate, or equatorial plate. It is clearly seen in the illustration that members of homologous pairs have exchanged genetic material. Crossing over accounts for producing genetic variation among gametes that will be formed later on. But aside from crossing over, the mere positioning of chromosomes along the equatorial plate during metaphase 1 could result to numerous possible combinations of chromosomes in the gametes that will be formed. To further elaborate on that point, let us take a look at the following illustrations. Let's say in our first case, for each chromosome pair, paternal chromosomes are aligned on one side of the metaphase plate, while the maternal chromosomes lie on the other side of the plate. At metaphase 2, a daughter cell contains paternal chromosomes only, while another cell only contains maternal chromosomes. Hence, upon completion of meiosis 2, cell 1 produces two gametes, each containing paternal chromosomes only, while cell 2 produces two gametes, each containing maternal chromosomes. In the end, 4 gametes were produced, 50% of which bears only paternal chromosomes, while the remaining 50% bears only maternal chromosomes. How would the resulting gametes look like, if chromosomes were aligned along the metaphase plate this way? The long chromosome, which is of paternal origin, and the short chromosome of maternal origin, lies on one side of the plate, while the long chromosome of maternal origin, and the short chromosome of paternal origin, occupy the other side of the plate. At metaphase 2, cell 1 will be having a long chromosome of paternal origin, and a short chromosome of maternal origin. On the other hand, cell 2 will be consisting of a long chromosome from paternal source, and a short chromosome from maternal source. These cells will be producing gametes, having copies of chromosomes, from both paternal and maternal sources. We have seen in the previous illustrations, that the random distribution of homologous chromosomes along the metaphase plate, could result to several possible combinations of chromosomes in the gametes that will be formed. Imagine how many possible combination of chromosomes, could be produced in this example, wherein members of homologous pairs have exchanged genetic material. After aligning themselves along the metaphase plate, kinetochore microtubules that adhere to the chromosome's centromere shorten, thereby causing the members of a homologous pair to separate, and be pulled towards the opposite poles of the cell. Take note that in anaphase 1, the cell is still diploid and has a chromosome number of 4. You might ask, how do we actually count the chromosomes? Do we count the number of chromatids, or the arms, or the centromeres? The answer is, we count the centromeres to indicate the number of chromosomes. So in this example, we've counted 1, 2, 3, 4 centromeres, so that means there are also 4 chromosomes in that cell. However, when the telophase stage was reached, and cleavage furrow has been formed to separate the two daughter cells, each cell has now haploid chromosome number. It is during telophase 1, that the chromosome number is being reduced into half. Now let us proceed to the chromosomal events, that take place during meiosis 2. Basically, meiosis 2 is entirely similar with the events that take place in mitosis. Please take note that we begin with a haploid chromosome number in each cell at prophase 2. The chromosome number is still haploid at metaphase 2. However, during anaphase 2, wherein the sister chromatids of a chromosome separate, and are pulled towards the opposite poles of the cell, the chromosome number becomes diploid. Remember that we count the number of centromeres as an indicator of the number of chromosomes within a cell. So let us count. And we have 1, 2, 3, 4 centromeres. But upon reaching the telophase 2 stage, and cytokinesis, each cell now bears only 2 chromosomes, and the haploid chromosome number is maintained. As what was mentioned earlier, meiosis II is also referred to as equational division, since the chromosome number at the start and end of the process was maintained. This time, let us compare the differences of meiotic events between male and female germ cells. Let us begin with that of sperm production in males. Sperm production in males is simple and straightforward. A spermatogonium will undergo meiosis I and II to form 4 sperm cells. On the other hand, the egg production in females is a bit complicated. An oogonia will undergo meiosis I and II to form only one mature egg or ovum. How did it happen? You might ask. Take a look at the following illustrations. There is an unequal distribution of cytoplasmic contents between the daughter cells of meiosis I. The smaller daughter cell is referred to as a polar body. In this case, it is specifically referred to as the first polar body. The larger cell and the first polar body will both proceed to meiosis II. The first polar body will divide into 2 polar bodies. While the larger cell will also divide, but the cytoplasmic contents of its daughter cells will be unequally distributed again. The smaller cell formed after the division is specifically referred to as the second polar body. The 3 polar bodies will degenerate later on, leaving only one mature egg or ovum at the completion of meiosis II. Aside from the unequal distribution of cytoplasmic contents between the daughter cells of each meiotic event, what renders egg production more complicated are arrest stages at some points of the development from oogonium to mature ovum. Activation factors such as hormones, and even fertilization is required for oocyte to resume, and to complete meiosis. The arrest stages and resumption of meiotic events will be discussed extensively in a separate video lesson on fertilization.